So the fourth presenter is Riley Macdonald from the University of Otago, supervised by Professor Amanda Black. And I'll just give you a title of your talk before we go. All right, so Phytophthora methods, are going to be presenting on Phytophthora methods.
Okay, sweet. So I was spending my time this summer working with Phytophthora agathidicida, which affects kauri trees. Yeah, I'll just jump into it. So why is this project important? Well, first of all, kauri trees are really important to New Zealand culture, to Māori, but also historically to our economy. Not so much anymore, but it was definitely a really big part of it when New Zealand was first, getting into the global market. But at the moment, the main strategies are kind of public awareness and these cleaning stations that you can see if you go up north to remove contaminated soil from visitors' shoes, which are both great, but they really depend quite heavily on public compliance, which is, variable at best and not exactly long-term systematic solutions. Because the detection methods at the moment are pretty unreliable and they actually take a really long time. Soil baiting is a big one, and the false negative rate for that is about 20 to 50%. It needs about 100 grams of soil for each test, and it takes, I think it was 15 to like 25 days to do the full pipeline from gathering the soil to getting a result. Soil kits as well are used pretty often, but inhibitors in environmental soil mean that only about kind of 20% of available DNA in the soil ends up getting extracted. So you get lots of false negative rates there as well. So our project was improving these current protocols, specifically with reliability, with speed, and with sensitivity, and also improving the ability to carry them out in the field to make hopefully a really community-based detection system. So, you can employ anyone to come and do these tests for you, not someone with like a lab background necessarily. So there were kind of two main technologies that I used over the summer. The first one are these cute little QIGen kits. So you can get them to target soil, so it's like directly soil or plant matter, which I believe they're normally used for leaves, but we also trialed them with roots. And so it comes with everything. It's got a bunch of steps. It takes maybe 40 minutes to run a test, and you end up with a little tube of extracted DNA. And the other one is qPCR, which is this little machine. It detects DNA and quantifies how much there is, and it uses specific primers, so you only detect, well, hopefully one specific species, which in this case was plant matter with an RBCL primer, which is just a gene that's in, I think, every vascular plant. It's pretty broad. So it's just a positive control checking for kauri, making sure everything's worked. And we're also checking for the actual PA. And so you get a little graph at the end of it. It looks like this. So in this run, we were testing different ways of handling the roots prior to carrying out the extraction. And so these numbers correlate to how many cycles it takes for the DNA to be detected. So the lower numbers equal more DNA, basically. Yeah. So our key conclusions. We had soil kits and plant kits, and we had soil and roots to test. So we tried all the different combinations of that. And we found actually the most reliable and I guess most user-friendly one was actually the roots with the soil kit. And then we added in a dilution step. So after you've used this kit and you've got your little tube of DNA, and before you put it in the qPCR to measure it, just a 1 in 10 dilution. And that's, I believe, used pretty commonly when working with plants because it's just hard to work with. It reduces the inhibitor down where it's not inhibiting qPCR anymore, but you're still getting DNA traces in the qPCR test. And so that had a massive effect in reducing false negatives. And then this was probably the biggest one. We redesigned the kauri qPCR primers. So like I said before, the positive test we were doing was quite general. You can use it with any plant. And so because of that, it's got quite a low temperature because they're kind of allowing for little mismatches depending on the species. And so we went in and we made a primer specifically for kauri, which meant that we could really increase that temperature. And that meant that it could match the Phytophthora primers at about 60, 61 degrees. And so when you increase the temperature up to here, we were getting way less false negatives with the PA. And we haven't done it yet, but Sanger sequencing is going to be done out to check for any non-specific amplification. But I believe if it was run, it would all be PA due to such a high temperature that we were using. Yeah, and when we combine all of this, so we did a full pipeline beginning to end, and we got this positive test. As you can see, the RBCL, so that's the kauri positive tests, they all showed up. That last column's a blank. And with the PA, there was only that one false negative with the NA there. So it was a really symptomatic sample taken from Northland. And then we ran a negative test, and yeah, no false positives. Again, all the controls worked. Yeah, I would say I think more of these tests need to be done to properly benchmark these, techniques that we've developed, but it's a really good indicator that we're on the right track. Yeah.
Awesome. Thanks, Riley. That's a really good talk. We have any questions for Riley? So, just checking through here. So, a question for you. Do you think you'll be able to upscale this as a multiplex type of interaction where you're looking for multiple Phytophthora species at the same time?
Well, I mean, all you have to do really is change the primers, which if you were doing it on a big scale, if you're testing a whole park, I think cost to like output would be quite reasonable.
Okay.
Because they're just little primers that and you can just use publicly available software to make them.
Yeah. What does
Um,
do you know what the price also works out to be for the individual kit samples for each
I haven't,
sample?
but I think that would be quite an interesting value to have. But yeah, I think the kits can do about 50 each, 100 each.